Bernhard will be Dr. Bernhard Hörmann. And Bernhard, together with his former PhD group from Professor Maja Kuhn from the University of Freiburg, they are actually some quite long-term collaborators from us. So we have worked together on quite a few years on several projects. And I think, yeah, the work with Bernhard is really one of the examples that show how well also this scientific collaboration between universities can work. By training, Bernhard is a biologist. He studied biology and molecular biology in Vienna. There he did his bachelor. Then he moved to the Ruprecht-Karls University in Heidelberg. There he did his master's. And then he started his PhD, which was also a collaborative PhD between the EMBL in Heidelberg and between the University of Freiburg. And in 2019, he defended and received his PhD. And now Bernhard finally did the big step away from academia and started working in industry. He works still in Freiburg and at the company Thermo Fisher, where he is working in the immune diagnostic laboratory. But the work that he will talk about today is still work from his PhD postdoc times in the group of Maja Kuhn. And Bernhard, we are super happy that you agreed on presenting this work, and we're looking forward to your presentation.
Okay, thank you, Tina. I'll take away your screen. Okay, can you see my screen in presentation mode now? Perfect. Yeah, thank you for the kind introduction. I've also deeply enjoyed our collaboration, and I know it's still ongoing with other people now, and that's why I'm presenting here. And since you have already kindly introduced our laboratory in Freiburg and the topic, and me as a person, I would directly jump towards the title. So our talk is presented MS-based approaches to study the substrate sequence determinants of phosphatases PP1 and PP2A. So, and the laboratory of Professor Maja Kuhn, where I did my PhD, is interested into studying phosphatases, and it does so by interdisciplinary approaches. So the lab consists of equal parts of chemists and biologists, and we have always closely collaborated in the lab as well as with people outside, and used chemical biology approaches to study phosphatases. And one focus of the lab is on serine threonine phosphatases, protein phosphatases to be precise, and there are two subgroups, one of which are the PPMs depicted here on the left for reasons of completeness, but there is a focus in the lab on phosphoprotein phosphatases on the right, and particularly on PP1 and PP2A, because together with the third one, PP2B or calcineurin, those are the really big players that regulate phosphoserine, phosphothreonine dephosphorylation reactions on proteins in the mammalian cells. These phosphatases are highly conserved, and I bet if you tell me your favorite pathway or the project you are working on, we could quite quickly find a link to one of these phosphatases. They counteract a subset of approximately 400 phosphoserine threonine kinases, and it is quite well known that they do that by actually choosing a completely different evolutionary solution than kinases, because they basically associate with regulatory proteins in a transient fashion, and thereby they form so-called holoenzymes, and then these holoenzymes are targeted to specific phosphosites where they counteract kinases. And what has not been in the focus of the research, because the level of the understanding the regulatory proteins is already so complex, is if the catalytic domain of PP1 and PP2A actually also contribute to the substrate selection to a certain degree. And this was in the main focus of my PhD. And in order to do so, we teamed up with two, we would say, world-class mass spec laboratories, one of which is the EMBL proteomic core facility in Heidelberg, and then with you guys here at BayBioMS in Munich. And I will present those two parts because what is already known is that here you see the structure of PP1 catalytic subunit on the left. There is some evidence that there are certain grooves directly. Okay, could all mute their mics? Thank you. So basically, there are grooves directly surrounding the catalytic pocket, depicted here by the two metal ions. And it is hypothesized that actually these grooves contribute to the peptide substrate selection in a combinatorial fashion. And also our lab has in the past, by bioinformatic analysis of quite small subsets of really well-defined phosphor substrate sites for PP1 and PP2A, identified that there is an enrichment of certain amino acids surrounding phosphoserine and threonine, and that this enrichment differs between PP1 and PP2A. Importantly, this was on the holoenzyme level. So these are substrates that have been identified together with the regulatory proteins. So when we really want to decipher the contribution of the amino acids surrounding phosphoserine and threonine, we would need to go to the peptide level, but peptide-based approaches were only carried out with quite small sets. Because whenever you go to quite large approaches using really large peptide libraries, you end up with quite laborious readouts, such as array-based setups or mass spectrometry as a readout, and that there's not too much out there. So we combined large phosphopeptide libraries with a comprehensive approach on the proteome level to confirm our findings. So on the peptide level, we designed an approach which we termed phosphopeptide library dephosphorylation followed by mass spectrometry, abbreviated PLDMS. And we designed libraries based on the biological insights we had. So we found that the 4 amino acids before the phosphoserine threonine are really important, and the 3 after. But of course, due to technical reasons, those libraries need to have a certain length. And what we did is basically we had 4 libraries which we termed N-term libraries, where we have these X1 positions, and I'll come to this in a moment. And we have 1 C-term library where we have this X2 position. So what does this mean? Basically, we want to have as many sequences as possible, but we still want to control complexity. So, and this X1 means that during the synthetic approach, during the synthesis of the library, we do not supply the reaction with a single amino acid which we want to couple, but with a whole mixture. And we chose 14 amino acids, again, based on biological insights, which we supplied in the reaction. And we always did this for 3 positions in 1 library so that the complexity is still under control. And by this, we can basically cover more than 25,000 different phosphopeptide sequences. And we still have an C-terminus, which is optimized for MS-based detection and readout. So what do I mean by an isokinetic mixture? If you want to have amino acids that are equally likely incorporated during synthesis, you actually cannot use equal parts, because due to the bulkiness and other biophysical properties, these have very different likelihoods to be incorporated. And that's what's meant by isokinetic. So actually you need to optimize such a mixture of amino acids over several rounds, as you can see on the right. And after several rounds, you have rates of incorporation that are sufficiently equal so that you have good coverage of all amino acids during MS. And you can really perform good statistic analysis in all of those. So the assay looked like this. We had this library, or one of these libraries that I've just introduced, in a tube. So this is in a single tube, a highly complex mixture. You incubate with the phosphatase, PP1 or PP2A, or you have an untreated control. And then you stop the reaction and you monitor this by a dephosphorylation assay with a pH shift, when basically the most preferred amino acids and peptides are dephosphorylated, but the reaction is not taken to completion. You do this by the addition of HCl. Then you again separate phosphatase from protein by means of size exclusion chromatography, and then you continue with the mass analysis. And by this, basically, we have 3 conditions. So 2 phosphatases, 1 control for the 4 N-term libraries and the 1 C-term library I've introduced. So this means 15 samples in total. And the cool thing is with a synthetic library, you know exactly what to put in. And for example, you completely overcome problems that you normally have with false localization rates of your post-translational modification and stuff. But you can really build your library-specific false discovery rates, and you know that the counts that you accept for your final analysis, that these are really of high quality. And basically from the 4 N-term libraries, we then had more than 350,000 data points, and for the C-term library, more than 80,000 that went into final analysis. So, and what did analysis tell us? So when we had a look at the incorporation rate over all peptides, we found that approximately 60% of incorporated amino acids in position 0, so where the phosphosite is, are phosphoserine, and approximately 40% phosphothreonine. But when we had a look at the ones which got dephosphorylated, we found that phosphothreonine was significantly more often dephosphorylated by PP1 as well as by the catalytic subunit of PP2A. And actually, we then could look into the library data, pick certain peptide sequences, synthesize them in a targeted fashion, and basically do a very basic biochemical dephosphorylation assay. And we could demonstrate that the same peptide, just differing in the amino acids, so in the phosphorylation side, phosphothreonine or phosphoserine, really makes a big difference, even in a non-competitive setup for both phosphatases. And the same logic you can basically apply in a more complex fashion to all amino acids surrounding the phosphosite. And what we found is, so what you see on the left is that when you do not only ask which are the amino acids which are more abundant in the dephosphorylated peptide compared to all the peptides, but you then again can divide the 2 phosphatases by each other, and this tells you actually which are the amino acids that are preferred by one phosphatase and not by the other. I will only show this data for reasons of time. So you see on the left that in blue, PP1 prefers the positive charges of arginine and lysine, whereas the negative charges are preferred by PP2A. And again, we could pick single peptides that just differ in these amino acids that are depicted here in bold. And we see that if you give only that amino acid, this peptide to the phosphatase, they are really dephosphorylated at significantly different rates. And the positive charges are preferred by PP1, and the scenario is the opposite for PP2A. So this is nice because on the phosphopeptide level, you can, and on the library level, you can really do good statistics and you can cover all amino acids at really good rates. But the question is, what's the impact of that on the protein level? And this is where we teamed up with the experts here in Munich. And so we then used phosphoproteomics in order to confirm that on the proteome level. And what we did is we took HeLa cells, incubated them with a small molecule inhibitor, caliculain A, a highly potent and also toxic inhibitor of PP1 and PP2A. And basically then we lysed the cells in order to really disrupt signaling complexes and pathways in order to only identify direct dephosphorylation sites for the phosphatases. We incubated the lysate with the recombinant catalytic subunit for a certain time again, and then we completely denatured all proteins by urea, went for TMT labeling and IMAC-MS readout. And what you see here on the right is when we have a look at class 1 phosphosites, so phosphosites that we have good confidence in, we see that phosphothreonine in light blue is significantly less abundant than phosphoserine, and this is something that is well known. But when we look at the sites that get dephosphorylated, both for PP1 and PP2A, threonine gets more and more enriched. So we also see that in a fully folded protein, again, there is no overlaying effects, but it's still a main effect that can be observed that these phosphatases prefer the phosphothreonine. As again, the same logic was applied to all the amino acids surrounding the phosphosite. And you can see on the left, again, positive charges, preference for PP1, and there's a slight preference for negative charges with PP2A. So basically this tells you, even if we have higher structures and highly complex proteins, this contributions that we observed on the library level still hold true to a certain degree. But such a heat map perfectly normalizes for the natural abundance of amino acids. And actually, if you would like to pick a follow-up, you would like to know, which are the amino acids that are most important in a biological context. And for this, we carried out motif analysis on the right, which is basically relying on raw counts. And we saw that the arginine in position -3 is most important. And this fits, first of all, with the preference we see for PP1. So we picked this motif for a follow-up. And it's also, if you're working on kinases or in the signaling field, these are motif characteristics that are well known from 14-3 and kinase motifs, PKA and PKB. And 14-3 proteins are signaling scaffold proteins that bind apart in a phosphodependent manner. And we then did a follow-up on that, but I won't show any details due to time constraints here. And we could also show by live cell microscopy that this is really of high importance, that these catalytic subunits detect those motifs. So my conclusions are, from a methodological perspective, we see that the development of a PLDMS method using those randomized peptide libraries is suited for being combined with MS-based analysis, and it's especially suitable for studying the sequence specificity of post-translational modification removing enzymes. And I stress removing enzymes, because if you want to study enzymes that remove a PTM, it's very difficult to encode those libraries by genetic means, because you need to have the PTM present as a starting point of a library. And I think the approach is especially powerful in that regard. And biologically, we see that PP1 and PP2A, as I've said, have this preference for phosphothreonine, and it's more and more, there's more and more data in the literature that this is, for example, really important during mitotic exit and all the dephosphorylation that happens there. And we see also that PP1 has an intrinsic preference for basic motifs. I don't have time to show structural data, but most likely due to the acidic groove that is directly surrounding the active site. This is not the case for PP2A, but this protein rather needs holoenzyme formation in order to also be able to dephosphorylate basic motifs. Yeah, and with this, I'm already at the end of my presentation. I would like to thank my former PI, Maya Köhn, for always really supporting that project and letting me carry out the thesis, Thomas and Dominic, who really contributed to the library part of this project. Of course, our great collaborators here in Munich. I deeply enjoyed this working together with you. And then the core facilities at Freiburg and Heidelberg, and you for your attention. And I don't know if there's time and space for questions now or later, but I hand back to you, Tina.
Bernhard, for the great talk. Yes, I think we definitely have time for a few questions. There may be some questions from the audience. I don't see anything in the chat, so please feel free and ask. Just unmute yourself and ask any question you might have. If nobody's brave enough to start, then Bernhard, maybe I just start. So for your experiments, it was super important to do these in vitro assays, right? Where you had the library and where you have your purified phosphatase that you throw in. How did you derive that purified phosphatase? And you said that usually this phosphatase works as a holoenzyme with other proteins. So how could you make sure that it's really the pure phosphatase and not when you purify it from some cells that there is also some holoenzyme interaction partners also co-purified?
Yeah, so basically, actually the purification of these phosphatases is pretty tricky in itself, because in bacterial expression systems, they are highly toxic because they dephosphorylate a lot of stuff. And so the PP2A, we could not purify ourselves, but we bought it from a company. And they did all the verification, run it on gels in order to see that it doesn't form higher complexes. And we also, again, did this before subjecting it to any experiments in our laboratory. And for PP1, our lab has done structural studies and also was able to crystallize it in the past. So we have good amounts of that. And also there we have good means, because also, for example, for crystallization, you need to make sure that you have really pure protein. And we made sure, for example, by co-expressing it with, or actually producing constructs that have larger tags and which are cleavable, that they don't associate with complexes in the bacterial expression system.
So actually not so easy, even though it sounds so easy to work with these purified proteins.
Yeah.
Cool. Any other questions from the audience? What do you think how does that impact now this whole concept of phosphatases that just retrieve their specificity by building different holoenzymes? How does that fit with it? So does the catalytic subunit just bring in an additional flavor of substrate specificity, or do you think that the phosphatase really sometimes acts completely by itself? How does that change this textbook picture of how phosphatase retrieve their sequence specificity?
I think basically that, yeah, as you say, it adds another level of complexity. And first of all, I think, yes, that it might contribute. So there are regulatory proteins that are basically just serving as a binding platform. And then basically you can imagine the phosphatase is brought towards the protein, but where to act on the protein, especially, I mean, in unstructured regions, I think there are these pockets, and the surface of the catalytic subunit might have a really big impact. And actually we have just published a review where we state that depending on the structure of the phosphosubstrate site, so whether the phosphosubstrate site is located in an intrinsically disordered region, where you just have this spaghetti loop that cannot add much of a template to a regulatory protein, then the amino acid sequence context might be more important than having a substrate site in a really rigid tertiary structure. And I think, yeah, this, as a scientist, we might not like it, but it's several levels of complexity and several fine-tuning steps that, depending on the structure and the holoenzyme and the size of the regulatory protein, can affect then which phosphosite is picked at very different levels.
Very much. So if there are no more questions from the audience, then I would pass on to Michi.
Yes.
Thank you. Thank you once more.
Thank you.
Thank you very much for the nice presentation. If there are no further questions, I'm happy to introduce our second speaker for today. This is Verena Peters. She is a food chemist by training, and she graduated here in Weinstefan. And after graduation, she joined the group of Thomas Hoffmann at the chair of food chemistry and molecular sensory science, where she started her work during her PhD project on MINT. And the part she is going to talk about today is also from her PhD project. She later stayed for a postdoctoral research stay at the chair. And today I'm very happy to introduce her and give her the stage. And I'm looking forward to your talk.
Thanks, Michael, for the nice introduction. So I have today the pleasure to lead you through my research, what I've done over the last few years in my PhD thesis. And there I focused on the development of a high throughput toolbox for a comprehensive flavor compound mapping in MINT. First, a few introductory words, what was the idea and the background, and why we focused especially on MINT. So genus Menther is an important member of the Lamiaceae family, and it is represented by about 19 species and 13 natural hybrids are known. Commercially, the most important MINT species are peppermint, Mentha piperita, spearmint, Mentha spicata, and Mentha avensis, the corn mint. The compounds that give peppermint the characteristic refreshing and cooling aroma, which we know maybe from toothpaste, for example, are menthol, menthone, and menthofuran. And the compound primarily responsible for the sweet candy cane, green and herbal flavor of spearmint, which we know maybe from chewing gum, are carvone, cineol, and limonene. In 2016, more than 42,000 metric tons of mint were produced, and it's rising from year to year because of its economic importance in the food, cosmetic, confectionery, and also in the pharmaceutical industries. And due to the economic importance, it is so incredibly important to secure a sustainable and robust supply of such an important plant for the industry. And a supply which is jeopardized through increasing demand for such a natural ingredient, through stagnant yields of the commercially important mints, of diseases and pests, and also the changing climate conditions. And due to that, properties and the characteristic of the aroma composition of a mint plant is also influenced by so many different factors, such as the taxonomy, the growing stages of the plant, and the climate conditions. And so the aim of my research was to enable high throughput quantification of the valuable aroma compounds in mint to map flavor alterations and to understand what's happening in the mint plant. To meet the new requirements, we had then to develop new methods. These methods had to be sensitive for all the key chemosensites and mint, which are already known in literature. We wanted to enable an analysis of hundreds of mint samples in a short time. And the method should be suitable not only for mint oils, but we wanted also to quantify mint leaves. And due to the high sample amount, the aim was also to use as less sample material as possible. Hofstetter et al. Could already establish a high throughput approach for the analysis of aroma and taste compounds in apple juice by means of 3-MPH, and that provided the basis for this research here. And in summary, the aim was that our approach should be faster than the traditional flavor approach and the GC analysis, and it should enable then the high throughput analysis of different mint samples to monitor chemosensate changes depending on factors such as the taxonomy, the harvest, the whole distillation process, growing stages, and also environmental conditions. And that brings me directly to the sample workup. I think everyone agrees with me that the sample workup is still the most time-consuming and labor-intensive step in the analytical process. And especially the traditional aroma approach for GC is very time and labor-intensive. It usually starts with a separation of the volatile fraction from the non-volatile fraction by, as I already mentioned, the solvent-assisted flavor extraction, or SDE, or for mint also steam distillation is often used. Therefore, you need a few grams of plant material to extract a sufficient quantity of aroma compounds for further GC analysis. Our new extraction approach for aroma compounds was based on a B-beta homogenizer. And it requires only a minimum of sample material of 300 milligrams, which is roughly the equivalent of 1 major mint leaf, and a solvent volume of just 1 milliliter. This proved to be enough material to identify and quantify the main aroma compounds in a huge variety of mentha species. For highly precise and accurate results, we used also the stable isotope dilution analysis, where we added the stable isotope labeled standards to mint leaves before the workup and the measurement. And a great advantage of this extraction approach was also that the extract could not only be used for the analysis of the aroma compounds, we could also use it for the analysis of polyphenols, pesticides, or other analytes of interest that can be analyzed by SEMS. Through the preliminary work of many research groups, the most important aroma compounds that make the order of mint so special and unique have already been decoded. The most aroma compounds are formed from the isopentenyl pyrophosphate via the characteristic terpene biosynthesis pathway, and make up a very high percentage of the aroma compounds which are present in mint. On the one hand, there are non-polar terpenes which do not have a functional group, here highlighted in green, as for example limonene, cineol, alpha and beta pinene. And through enzymatic formation, these terpenes are then formed into carbonyl compounds, here in red, like carvone, polygol, and mentone, and also in alcohols like the mentone and linalool, which are representing then the third group of analytes. However, the aroma diversity of the genus Mentha is not only formed via the terpene biosynthesis pathway. Compound classes such as sesquiterpenes, as the alpha humulene and transcaripholene, and acids are then complete then the comprehensive flavor impression of mint. For analytics, it is not always the chemical similarity of the compounds that is important to ensure a chromatographic separation and detection. Derivatizable functional groups are even greater or are of greater relevance to adjust the analyte property of such highly volatile aroma compounds to perform LC-MS analysis and to enable liquid chromatographic separations. Also for the improvement of the detection and also to enhance the sensitivity. By subdivision of the 59 selected analytes according to their functional groups, there were mainly the carbonyl compounds, here in orange, like hexanal, for example, polygone, carvone, and alcohols highlighted here in green, where we have, for example, the menthol, 3-hexanol, and at least we have the terpenes, which do not have a functional group, as I already mentioned, for example, the limonene, terpinolene, alpha and beta pinene. So we had 59 different compounds for which a liquid chromatographic separation and a detection approach had to be developed in order to characterize then the aroma profiles in different mint species. And that brings me already to the first group where we had to develop a new approach, the carbonyl compounds, to make the volatile carbonyl compounds like the hexanal, mentone, and carvone more suitable for the LC-MS analysis. A derivatization step by 3-MPH, so 3-nitrophenylhydrazine, was required, and to ensure an LC-MS analysis. Therefore, we used a volume of just 40 microliters of extract or diluted mint oil, added 20 microliters of 3-MPH and EDC, and incubated it for 30 minutes at 40 degrees. And after a dilution step, it could be directly used for an LC-MS analysis. Through the use of the stable isotope dilution analysis with stable isotope labeled molecules as internal standard, workup losses were considered based on a molar ratio. And due to the limited commercial availability of some stable isotope labeled standards, for some compounds, an isotopically compound similar and structured was used for the quantification purpose. As for example, stable isotope labeled 3-methylbutanol was used for the quantification of 3-methylbutanol, 2-methylbutanol, and also isovalerate. After this derivatization step by 3-MPH and optimization of the MS parameters, highly sensitive MS detection over a large concentration range could be achieved for in total 19 different carbonyl compounds. Within 10 minutes, we were able to separate in total 19 compounds on the C18 column, which you can see here. And we were also able to separate isomers as, for example, the alpha and beta ionol and the already mentioned 3-methyl and 2-butanol. Using the derivatization reagent glycidyl-3-methylammonium chloride, so GTMA, we could modify the active hydrogens of the hydroxy groups of the alcohols to ensure an LC-MS analysis. For the detection and quantification of these alcohols in mint oils or the mint extracts, a derivatization time of 2 hours at 55 degrees was fully adequate to modify primary, secondary, and also tertiary alcohol present in mint. And also here for the precise quantification results, we used stable isotope labeled standards, which are highlighted here in blue. In contrast to the separation of the carbonyl compounds on a standard C18 column, a pentafluorophenyl phase was suitable for the separation and analysis of the GTMA modified alcohols. The mechanism of the interaction and separation is based on PP interaction, electrostatic and ion exchange interaction, and also hydrogen bondings. And within 12 minutes, we were then able to separate and detect 26 different alcohols, and we could also quantify them by the stable isotope dilution analysis. In comparison to the aldehydes, ketones, and alcohols, the terpenes are non-polar compounds which do not have any functional group for a suitable derivatization. Therefore, we used atmospheric pressure chemical ionization, so APCI, for the determination of this compound class. And the APCI makes the LC-MS more suitable for mainly low and medium polarity compounds. At the beginning, the MOBA phase and the analytes are first nebulized and vaporized by heating, and then the resulting vapor and solvent molecules are ionized by corona discharge. And so, and then stable reaction ions are generated. And at last step, the protons are then transferred to the sample molecules, and we can so detect non-polar compounds as the terpenes. As mentioned previously, for highly precise and accurate results, the quantitation of the terpenes was also performed by stable isotope dilution analysis using internal standards added to mint leaves or to the mint oils prior to sample workup and measurement. And since the monoterpenes are formed of isoprene, so a C5 unit biosynthetically, the chemical structure are very similar, and also they form then identical pseudomolecular ions. But through appropriate chromatographic, and so the appropriate chromatographic separation was essential. With a C18 column, we were able to separate not only these seven very similar compounds as the camphene, sabinine, myrcene, beta-pinene, limonene, terpenoidine, and alpha-pinene. We could also separate 12 further terpenes, and we could quantify them within 50 minutes using atmospheric pressure chemical ionization. Then to assure the quality of the methods, of the 3 developed methods, the validation of the was an important aspect. First of all, the recovery rates were calculated for the 2 different sample types, so for mint oils and the mint leaves. Therefore, we spiked 3 different concentration levels to the sample material. And for mint oils, we used, for example, analyte-free medium chain triglycerides. And for all the 59 compounds, they were in a range of 80 to 120%. As next step, we also determined the limit of detection and the limit of quantitation for all the 59 compounds, and for the, which you can see in this figure. For the carbonyl compounds, the LODs and LOQs range from 0.2 nanomole per liter to 0.8 micromole per liter, for example. For the terpenes, they were a little bit higher compared to the other compound classes due to a lower detector response. At least we also we compared the LOQ values to the order thresholds of the analytes to get an insight how sensitive our methods are, and therefore 26 out of the 59 compounds, for example, hexanal, 2-phenylethanol, and alpha-humulene, could be quantified by means of LC-MS below the order threshold. And for 11 further compounds like alpha-ionol, jasmone, they were kind of the same range like the threshold. But at this point, it must be noted that all the 59 aroma compounds could be analyzed without any enrichment step of the extracts, and the mint oils had to be diluted to ensure an analysis by means of LC-MS. To conclude the method development, it can be summarized that this toolbox enabled a high throughput analysis of hundreds of mint samples in a reasonable period of time due to the very fast sample workup and due to the low amount of sample material needed. And we could direct measure concentrations of 6 orders of magnitude in one run without any further injection of different sample dilutions, for example. And at least a big benefit of this method is also that the extract could not only be used for the analysis of aroma compounds, but also for the analysis of polyphenols, pesticides, or also for untargeted screening. So this toolbox perfectly meets the requirement to analyze hundreds of different mint samples. And like we could analyze different mint oils and different growing stages, we analyzed field samples, also different mint varieties, and also had a look into the environmental condition, what impact do they have on the aroma profiles. But due to the limit time, I will go into one sample set in more detail, which I think particularly highlights the benefits of the developed toolbox. So the motivation for developing this new extraction approach for volatile aroma compounds in combination with modern LC-MS methods was to characterize the aroma composition of each mint species by the analysis of one single mint leaf in order to avoid the traditional, crucial, and as well as time and cost-intensive distillation step of the mint oils prior to the analysis. And this toolbox enabled for the first time an analysis and a comparison of the aroma composition of mint leaves and mint oils of several species, as shown here in this heat map for black midgem, which is a peppermint, Mentha arvensis, and also 2 different spearmint types, so native and Scotch spearmint. And as you can see in this heat map and in the PCA here on the right side, the differences in the aroma composition cannot be greater. We were able to analyze 59 aroma compounds in mint leaves and also in mint oils. So here you can see the mint leaves, and here the mint oils. And we were able to analyze these compounds in one batch over a concentration range of 6 orders of magnitude without injection of different dilutions. And with these new developed methods, analysis with a linearity over large concentration ranges could be achieved. So we were able to quantify carbonyl compounds from 0.2 microgram per milliliter, which you can see here. For example, for isovalerate, over 200 micrograms per milliliter for carbonyl compounds such as hexanal or octanol, up to 400,000 micrograms per milliliter here, for example, for menthol in the peppermint oils. And yeah, as I already mentioned, this is only a small insight into what we have analyzed within this toolbox, and it has also been applied for to map the flavor alterations depending on environmental conditions and also growing stages. And that brings me already to my summary. So we could develop an extraction approach for one single mint leaf by means of a beta homogenizer. We could then develop 3 different stable isotope dilution LC-MS methods for an accurate and precise quantification of aroma compounds in mint leaves and also in mint oils over a concentration range of 6 orders of magnitude. We could verify the functionality of the methods by several validation experiments, and we could then use this toolbox to map flavor alterations. And so we could also support, or with this toolbox, we could support several breeding programs to sustain mint as a valuable natural product for the industry. And at least I want to thank you for your attention. And if you are interested in the developed methods in more detail, you are welcome to read all about my research in a recently published paper. And in a few weeks, there will hopefully also a second publication, which is focusing on the application of the toolbox and the mapping of flavor profile changes, depending on different factors. And at least I want to thank you. I want to thank everyone who contributed to this research, to all my cooperation partners, and also to the chair who made the research possible. So thanks for your attention.
Yes, thank you very much, Verena, for this very nice talk and of course of your very comprehensive mint analysis. Are there any questions for Verena? You can just raise your hand or unmute yourself, or also use the chat function. I don't see anybody so far, so maybe I will go ahead and start. I have one question regarding the alcohol derivatization. You said that you could derivatize primary, secondary, and tertiary alcohols alike. I was wondering if it was necessary to use the exact isotopically labeled standard for each type of alcohol, or if you could also get away with just using a structurally similar alcohol for the derivatization.
Yeah, so we checked that. So for the primary alcohols, the derivatization worked pretty good. And also we could see the derivatization time of 30 minutes was also enough already to detect, to modify all these alcohols. But especially for the tertiary alcohol, like the alpha-terpineol, yeah, therefore it's better that you have also a tertiary alcohol or isotope labeled tertiary alcohol to get better results in the recoveries. But I think for the 26 different alcohols, we used, I think, 10 or 12, when I remember rightly, isotope labeled standards, and the recovery rates were really good.
Okay, thank you. This was just I was just wondering if the reactivity of the different alcohol species was significant, but this is really, really nice. I also just wanted to ask about you showed that you could already quantify most of the odorants of most of your compounds in concentrations below their odor threshold. The compounds that you couldn't detect below the odor threshold, are they specifically relevant, or are they very important, or are they just minor compounds that you could get away with?
Yeah, so I think under the threshold there were only 10 compounds underneath the threshold.
Okay.
But therefore we have to say that, for example, for the mint oils, we diluted them 1 to 7,500 to perform the LC-MS analysis. So I think that's, yeah, not a really issue that the LOQ was beneath the threshold. Okay, so in case this would be relevant, you could just step down one dilution step and then just quantify everything below. Okay, this is really, really nice. Are there any questions from the audience? Just going to look through the list.
I was going to ask a question, Verena. Where do you see the future of this method? So I can imagine that for the industry, it would, for example, be super cool if it would be possible to determine the quality of a mint sample like this. You showed in your one application that you do see this super good separation, but I thought it was different MINT types, right? What's your feeling? Do you think, will it be possible based on these compounds to say whether this is a very good or a less good quality MINT package? And is this something that you envision?
Yeah, so they're already using this method in the industry for quality checks, of course, to see what's the concentration is like when they have MINT oils, because it's much more faster than the GC analysis. And what it's definitely used for breeding programs. So when you do the breeding, because yeah, we need new MINT cultivars that have aroma profiles like the commercial ones, like the peppermint. So the aim is to get a new breed that has the aroma profiles like a peppermint. And therefore, yeah, it's much more easier because you need only one MINT leaf, and then you can directly use it from the greenhouse and can directly analyze it without doing the growing on the field and then the distillation process. So it's much more faster to already see the aroma profile, for example, in breeding programs.
Great. So if there are no more questions from the audience, then thanks a lot to Verena for the presentation. And we quickly move to our last speaker of today, which will be Zachary Arden. I hope I pronounce your last name correctly, Zachary. You must complain if not. So Zachary is currently a postdoc at the very famous Wellcome Sanger Institute in Cambridge in the UK. And there he works on bacterial evolutionary genomics. But before he started in Cambridge, he actually did a postdoc at the TUM, at the TU Munich, and that's also where we met and where we started collaborating. And Zachary also did a short postdoc at the Karlsruhe Institute of Technology. And his PhD he did on the other side of the world in New Zealand at the University of Auckland. And also there, his topic was microbial experimental evolution. So microbial evolution follows Zachary through all his scientific career, I think. And that's also the topic that we will talk about right now. So Zachary, thanks a lot for being here and for presenting to us.
Thank you very much, Tina, and thanks everyone for coming. This is a very different talk, but hopefully interesting. I'm mostly an evolutionary biologist, and that's the angle that I'll be presenting this from. So detecting new genes in bacteria and viruses, the challenges and opportunities of doing that. Well, the subtitle is Got to Catch 'em All for proteins, even if they're short, overlapping, or taxonomically restricted. And I'll discuss what these things mean and why this is interesting and important. Firstly, just thanks to various people who are involved in this, including Tina and Chen at BayBioMS, and many students and collaborators in other places. What do I mean by new genes? So it used to be thought that the way you could get a new protein or new protein coding gene was only by a method which is called duplication and divergence. So in other words, by copying a gene and modifying an old one. So it's illustrated here. We have an original gene. At some point, it gets copied. So somewhere else in the genome, there's another copy. And then over time, the other copy changes maybe faster than the original version. So this is the standard view of gene evolution. And then people would say, well, that's the way you get a new gene. How did the green gene arise? Well, originally it came from the blue gene. This is still true for many genes. However, genome data has changed the picture because many genes show no detectable similarity to previously known genes. So you can imagine there's a red gene here that's not at all similar to the blue gene. Where did that one come from? And various evolutionary studies in the last few years have shown that some of these protein coding sequences evolved quite recently, ultimately from non-coding sequences. And for me as an evolutionary biologist, this raises a lot of interesting questions. But the first task, which I'm focusing on today, is how do we detect these genes? So the two challenges I'll be focusing on, firstly, that genome annotation is not a solved problem. A lot of people think it is, but I'll explain why it's not. So even in bacteria and viruses, which should be the easiest and which we think we understand quite well, we still actually haven't fully solved the problem of genome annotation. And secondly, that standard mass spectrometry methods don't capture all the proteins. So the first challenge is that genome annotation is not solved. Now, in response to this, a lot of people would think, well, don't we just pick the long open reading frames and call these genes? And this is basically the standard method of gene annotation. And it's illustrated here that you have different possible reading frames, ATG. And then if you shift it by one, you get a different amino acid, a different codon, a different amino acid sequence. And generally, we would pick this long one, say that's probably a gene. And that's a method that works reasonably well. And we just exclude the other ones and say, because there's already a gene in this region is covered, and we move on to the next region in the sequence and try and pick any genes there. So that's the standard method. And it makes sense. In various ways, it makes sense, for instance, to expect that if you have a long open reading frame, that it would be protein coding. That's basically because there's 64 possible codons, 64 possible genetic triplets, and 3 of them are stop codons. So approximately 1 out of 20 codons are a stop codon. So roughly, if you have 20 codons in a row, if you had a random sequence, you would expect approximately 1 of them would be a stop codon. So if you have like 100 codons in a row and none of them are stop codons, that's a good suggestion that probably is a gene. So that's why picking long open reading frames makes sense for finding genes, but it doesn't help us with short proteins, and it doesn't help us if the genes significantly overlap. And that's basically, very simplistically, that's why short proteins and overlapping genes have been missed until fairly recently. No, so the recent results are that there are lots of short proteins, and just in the last few months, these are some of the papers, and there's, there's I guess a new paper in this area every week or something, showing for different species of bacteria, in this case, there's proteins which had been missed previously. So this is using a method called ribosome profiling, which I'll briefly discuss. And this paper is mostly proteomics. And this was one of our papers, also mostly using ribosome profiling and a general review of the topic. Okay, so the second challenge is that standard methods of mass spectrometry won't help very much. And that's, this is obviously not my area of expertise, so Tina can probably say a lot more about this, but I'll just briefly summarize why I think this is the case. So mass spectrometry methods basically require that there's a sufficient amount of detectable peptides. Some short proteins just won't produce the right kind of fragments. So in principle, they're not able to be detected. Some proteins might have unusual chemical properties, which means that they don't have the kind of patterns that we expect. Many are weakly expressed and hard to detect with any method that's based on expression. They might only be expressed under particular environmental conditions or just very weakly, like maybe just 1 copy per cell or something like that. And that's going to be very hard for any method to detect. And many of them are unannotated in the first place, so that we're not looking for them with the kind of standard pipelines, which assume that the annotation is more or less correct. So what can we do about this challenge? And the basic answer that I'll present today is that using multiple methods can help, and I'll present 2 case studies from our collaborations in Munich. First project is from SARS-CoV-2, looking into SARS-CoV-2, and the second is looking into a respiratory bacterial pathogen, Pseudomonas Aeruginosa. Okay, so what about this idea of overlapping genes, which I've briefly mentioned before? What does that even mean? What is an overlapping gene? So this is one of the very important proteins in SARS-CoV-2, the nucleocapsid gene N. I think it's the most highly expressed protein, and it's actually used for detecting whether you have SARS-CoV-2 or not, partly probably because it's so highly expressed. So this is part of the sequence, the genetic sequence, which is turned into amino acids as shown there. And this is the part of the tertiary structure. But it turns out that in exactly the same genetic sequence, there's actually a different protein that's also encoded, and that's illustrated here. So you'll see it's a different set of a different series of amino acids, which is actually encoded by the same sequence and what's called a different reading frame. So if you put them together, the same stretch of RNA is encoding two separate protein molecules. Both form what we would classically regard as a protein, tertiary structure, and everything. And this one is also reasonably highly expressed. It's called ORF9b. This is discussed in a kind of a blog post that we put out a few weeks ago. Okay, this is the context of the SARS-CoV-2 genome. The genome, as you probably know, is around 30,000 nucleotides. And the part that we're interested in for today is the last part, which encodes the structural proteins in blue and the accessory proteins in green. And these other less well known about proteins in red, the overlapping proteins, the overlapping genes which encode overlapping proteins. So this is SARS-CoV-2, but before, when SARS-CoV-2 first was discovered, the most famous comparison that we had was SARS or SARS-CoV. And this is what the SARS genome looks like, very similar. As you can see, it has the same structural proteins in blue. It has basically the same accessory proteins in green, apart from during the SARS outbreak, there was a break in one of the proteins, ORF8. So it's basically the same. Also, the overlapping genes are largely the same, but actually this one, ORF3C, was not known about when the SARS outbreak happened, and no one discovered it until recently re-looking at the genome thanks to SARS-CoV-2. This gene, ORF3D, doesn't even exist in SARS. So there's something roughly similar there, but it's not encoded as a protein. The stop codons are blocking translation of the reading frame. So it's very similar genetically, but still there are some differences in the proteins. And unless you did a really careful study of the genome, you wouldn't even know that these proteins existed. So some of these were not, so particularly this one, ORF3C, was not even discovered. Even though it was there the whole time, it wasn't discovered until SARS-CoV-2, when people really started looking very carefully at the genome. And this is the region that is interesting for the studies that we did on this topic, which is the region overlapping this gene, ORF3A. So the methods, quickly, mass spectrometry was able to detect just one of the overlapping genes, the most highly expressed ORF9b, which is reasonably well, it's conserved throughout related viruses, and it's reasonably well known, but it's still not as well known as it should be. So a lot of genome annotations still don't include ORF9b. So this one is definitely expressed according to mass spectrometry, but the other ones aren't. But they do have evidence of expression from other methods, particularly ribosome profiling, which I'll discuss next, and from the thing that I focus on more, which is evolutionary conservation. And these things are discussed in this paper that was published in eLife last year. Okay, so what is ribosome profiling? Basically, it's a very special version of RNA sequencing. And it's a kind of RNA sequencing that finds the RNA that's covered by ribosomes. So firstly, you store the ribosomes in the cell with some kind of drug. There's different, or cook with cold, there's different options. You harvest the cells, you lyse the cells, you digest away, using enzymes, you digest away the excess RNA that's not protected by the ribosomes in blue. And then you're left with just little, very short segments of RNA that are protected by ribosomes. You can pull out the ribosomes and sequence the RNA, and then this is ribosome profiling. Very quick. So with that, we're able to detect some of these genes that I mentioned, that there's some evidence of expression of these different genes according to this method of ribosome profiling. Another case study in which we used this, which illustrates it nicely, is this bacterium, Pseudomonas Aeruginosa. And we found a couple of very long overlapping genes in this bacterium. In this case, the overlapping genes were on the antisense to genes that we already knew about. So in this case, there's a long gene here, TL8E3, which is annotated in the genome. It's understood what its function is. But with this method of ribosome profiling and RNA-seq, we were able to discover this other open reading frame on the other strand, in this case, the plus strand, that is also expressed. We're then able to follow up with mass spectrometry and show that there are actually peptides which are able to be detected as well. So in other words, we show that there's transcription, there's translation, and the translation is verified with peptides through various methods that are discussed in the preprint, which is available on bioRxiv. That was the first method, ribosome profiling. There's a second kind of class of methods, which is the evolutionary methods. And this, just very basically, the line that we're interested in here is the yellow line. And basically, over the, this is the same region, just in the other direction. Over the overlapping gene, there's a reduction in this yellow line. There's a decrease in the amount of diversity that's happening in this region. So in other words, there's this region is constrained, the sequence, more than you would expect, given the sequence as it evolves in other bacteria that don't have this overlapping gene. So that's the white line. So the white line is what you would expect if nothing interesting is happening here. And because the yellow line tends to be under that, that shows a sequence constraint and shows that there seems to be something functional happening in the region of this possible overlapping gene. So this is another class of evidence showing that there seems to be a protein being encoded here, which helps to verify or corroborate the evidence from expression that we see from ribosome profiling and mass spectrometry. Okay, so this was a very quick overview of this whole field of short proteins and new proteins. What can we conclude from this overview? Firstly, a really simple point, but I think it's important, that I wanted to say is that biology is often not about proof and disproof. Instead, it's very statistical. It's about probability. And often we need to rely on imperfect evidence in a cumulative case of multiple strands rather than a simple proof or disproof. So you could say that something is disproven, means the chance that it's true is basically 0%. What's proven, the chance that it's true is basically 100%. But actually biology usually happens somewhere in the middle, or a lot of interesting biology happens in this middle area where we haven't proven something, but we have some reasonable evidence for it. And I would say that the whole field of small proteins, to various extents, is somewhere in this middle section. For some things like this overlapping gene that I just talked about in Pseudomonas Aeruginosa, we have multiple strands of evidence. So I'd say it's very clear that there's something here that seems to be expressed as a protein. But for some others, we only have one strand of evidence. And it becomes weaker, but there's still some evidence for it. So I think it's helpful to think statistically about these things and not just is it proven or disproven. Second conclusion is that genomes are highly dynamic when you compare different strains and species. So for instance, different viruses like SARS and SARS-CoV-2. So SARS-CoV-1 and SARS-CoV-2 are very similar, something like 90% similar at the nucleotide level, but they still were different in the genes that they encode. So genomes are highly dynamic. They're evolving very fast, especially the accessory genes and viruses. They're also very complex. There's a lot happening there that we don't understand yet. And even in very well-studied microbial genomes, like pathogens, which have got a lot of attention on them, we still keep finding new genes and new things that are happening in the genome. The last conclusion that I want to, this argument I want to make is that genome annotation should take in information from multiple angles, including proteomics and evolutionary data. So I would say that understanding the fundamental biology, for instance, the structure of the genetic code, which might seem very theoretical, very abstract, and also even the fundamental evolutionary theory, like gene origins and the patterns of molecular evolution, things like the graph that I showed that showed an increase in sequence constraint, all of these very theoretical things can be really helpful in very kind of applied tasks that might seem straightforward, like genome annotation. So again, thanks to the various people who were collaborating on this. If you have any questions, I'm very happy to take them. I'm very happy to give comments if you're involved in gene annotation. And yeah, thanks for your attention.
Thank you, Zachary. What a great, almost philosophical presentation here. And I'm super happy we have a question from the audience. Farah, you're welcome.
Yeah, hi, thank you for this lovely talk. In Arabidopsis, we were alerted to the fact that we've been missing small peptides by the genetics. So we had, for instance, clavata or peptides that had very strong phenotypic effects. And it took a while to understand that we were missing the gene because we didn't see it because it was so small. And my question to you is, in your analysis of model organisms, or to what extent you've taken genetics and model organisms into account in terms of discovery and coverage of these small peptides?
Yeah, thanks. So are you thinking in terms of like functional genetics, like
Functional genetics, forward genetic approaches, etc. Mainly forward genetic approaches in model organisms.
Yeah, so I'm really not an expert in this, but we have done some of this, and this, yeah. So this paper here, this includes some like overexpression analysis, and this is the kind of thing that Professor Scherer's group was doing for many years, is individually looking at genes and either doing knockouts or overexpression in different strains, mostly in pathogenic E. coli, and showing that there is a phenotype. So I think that's the kind of thing you're getting at.
I'm getting at forward genetics. So in Arabidopsis, we had one of the very first higher organism genomes in the year 2000, right? And when we annotated the genome, we realized very much what you were saying at the beginning of your talk. We completely underestimated the very short peptides as being ORFs and as being basically functional units, you see. And this is where our tradition of forward genetics helped us see that they were associated with very strong phenotypes. And that's how we reannotated the genome based on the forward genetics. So it's not exactly going out there with a reverse genetic approach and overexpressing or knocking out genes in a systematic way. Do you see? It's more just a forward genetic approach, which was the tradition in our field since the 80s. And the same would have applied to Drosophila or to Cinorhabditis. Do you see where I'm coming from?
Not really. And terms of, I don't actually remember the methods of forward genetics. But basically these things have been kind of dissected in various ways. And so they were originally found through RNA-seq, that there was some kind of RNA being expressed. And then that was verified with Ribosome that this RNA is not just expressed, but actually translated into proteins. Occasionally with mass spectrometry as well. So that was kind of the starting point, and then they were dissected with, I guess what you'd say is the reverse genetics of knockouts and overexpression. Probably someone could say something more sensible about forward genetics, but yeah.
Thank you.
Bernhard, go for it.
Thank you for the nice talk. So so you brought up this nice example with the SARS-CoV-2 gene, where actually there's another smaller gene with an ATG coming directly after, right? Yeah, exactly that
Right.
one. So I would imagine, because if the ribosome is scanning and is supposed to pick an ATG, as far as I recall, this is determined by in mammalian, it's the COSAC sequence, right? Which determines if the ribosome really picks that ATG. So is there any evidence, or do you have any insights that long known proteins with reasonably good but not excellent COSAC sequences in mammals or their equivalents in bacteria are more likely to harbor those small peptides or small proteins you're investigating?
Yeah, so that's a great question, and I don't know the answer to that. So we haven't, like, I've thought about similar things, how to what kinds of genes are overrepresented in terms of frame shifts or in terms of overlap encoding, and I haven't so far found anything about that. But I guess you could invent hypotheses in either direction. You could think that if there's a strong signal there, then there's less likely to be overlap encoding. Or you could think if there's more ribosomes binding, then you're more likely to accidentally get some kind of slippage and more likely to overlap encoding.
Yeah, I guess it also depends. I mean, there are these effects, I think, which are pretty well described, of basically ribosome traffic jams, right? If the sequence is too good, so then you think that there could also be a scenario where actually ribosomes start jumping a good ATG due to that.
Yeah, so in this case, the ATG of the N protein is just like, I think, 9 nucleotides upstream, but sometimes it's well downstream. So I haven't found kind of standard rules, but it could be, it's quite possible that there's some kind of RNA element that assists with the jump, but I haven't been able to find anything. Like, there's a few individual cases that have been shown, but I don't think anyone's kind of got a general theory of how this happens. A lot of people discard most of the stuff as just like noise, But in this case, I think it's clear that this is a nice protein, and there's a bunch of functional studies that have been done on these things. But a lot of this may just be kind of accidental slippage of ribosomes. It's still a very open question how this is happening and whether it's functional and how much is functional and that kind of thing.
Okay, thank you.
Any other questions from the audience? I mean, I find these overlapping genes absolutely fascinating. I still think it's crazy that this is happening. And when I think about it from an evolutionary point of view, I always think that somehow these two genes that are coded by the same sequence with the frame shift must have been developed together, right? Because it is very unlikely that, you know, you generate a protein that is optimized for something by a frame shift. So you already said, Zachary, that your way to look whether, so to me, it makes lots of sense that you will have less mutations in a gene sequence which codes for two proteins, because if any mutation happens, maybe, and both proteins are important, right? It is less likely that this will be good for the cell, because maybe a mutation in the one gene is bad for the other, for the one protein is bad for the other. So that makes lots of sense. But how do you at all explain how these things happen, that you in SARS-CoV-2 suddenly have an additional protein that doesn't exist in SARS-CoV-1? How likely is it that this additional overlapping gene has a function and is something meaningful? It must have evolved a long time before. That would my belly feeling be. How do you explain that?
Yeah, that's a great question. And that's why, basically, I think that's why people discount it as, well, okay, if it's there, it must just be noise, it must be junk, because, you know, what are the chances of it being functional? But it depends. So one thing that we showed in this eLife paper is that there's some tendency for that the hydrophobicity of the protein that's produced, the ORF3D, the ORF3D protein has a very similar hydrophobicity to, so this one here in yellow has very similar hydrophobicity to the ORF3A protein. So that suggests there's some tendency, at least, for like structural, that maybe it has a kind of a similar structural elements between ORF3D and ORF3A. So that's a property of the genetic code, that a frame shift actually tends to preserve hydrophobicity. So the hydrophobicity of an amino acid, and then what it frame shifts to, tends to be relatively similar, much more similar than you'd expect by chance. So I would say the code seems to be set up in such a way that frame shifts are less destructive than you might expect. So that's part of the story. But probably most of this that happens is just junk and then it's lost. But some of it isn't and gets conserved. And it's probably, I mean, statistically, you'd expect it's the functions that are quite easy to get. Like maybe it's just helpful for the virus or for the bacteria to have a small hydrophobic protein around for some reason, that improves some simple aspect of function, and then that might be conserved. Yeah, so that's my hypothesis, that it's kind of simple functions that are quite easy to find. There's some tendency in the code to preserve structure, and those things together allow, sometimes you get a functional protein.
Philosophy, I said it already. Very cool. Okay, super. Thanks a lot, Zachary. Are there any final questions from the audience? If not, then I'm very happy to conclude this seminar. Big thanks once more to all our three speakers, and I hope I will see many of you again in February. Have a nice evening. Ciao.